Okay, thank you for coming. I will introduce the speaker of today, Dr. Gustavo Garcia Gomez-Tejedor. He did his PhD at the physics department of the Complutense University in '78, and then he was assistant professor at this university from '82 to '87. He was, after that, staff scientist at the CIEMAT from '78 to 2002. And from that year on, he was a research scientist at IFF-CSIC. In addition, he's also appointed professor at the Center for Medical Radiation Physics at the University of Wollongong in Australia since 2009. And here he has been the PI of several national projects and the Spanish representative of over 12 international projects, Marie Curie networks, cost actions, etc. He is co-author of over 290 scientific publications, and he has been supervisor of 12 PhD students. So I think his presentation will be of interest for you because it has very important applications in medicine, as you will see, and an important dose of good physics. So thank you.
Thank you. Can you hear me? Thank you very much for this introduction and for inviting me to give this seminar, in which we are going to discuss, to present some applications, medical application of radiation, and to see an overview of some method that we can use to convert the radiation damage into radiation benefit. As everybody knows that the medical application of radiation started in we can say 1895 with Röntgen's discovery. He was studying the effect of these cathodic rays, electrons, when he realized that some new radiation came out of the tube and was a special radiation because it was able to cross through some bodies, some plates, even relatively thick plates, and it was possible to blacken the photographic plates. And this, you can see, this is what we can consider the first radiography, which is the hand of Röntgen's wife. She died of cancer, as you know. This is a very simple machine. I have some, we can construct a machine like this because it's quite easy. It's simply, you produce some electrons with a filament for thermoionic effect, and you accelerate the electron with a voltage, apply a negative voltage to the cathode, and then this accelerated electron, you stop them with a relatively heavy metal, like tungsten. So the deceleration of the electron is producing, as you know, radiation, electromagnetic radiation, which are the X-rays. Then, only 1 year later, Becquerel discovered the radioactivity, as you know. And he found other kind of radiation very similar to Röntgen radiation, but coming from some materials. In his case, he was working with uranium salts, and this material produced similar rays that initially were called U rays because were coming from the uranium. Lately, with the works of Marie Curie and other researchers, were identified. So this was possible to blanken the photographic plates at their X-ray, but later we identified a particle beams, alpha radiation that you know is formed by atoms of helium. beta radiation, mainly electrons, initially electrons, and gamma rays. Gamma rays are similar to X-rays, but the origin is different. The X-rays is coming from the electron, free electrons or electrons on the atom, on the core of the atom, and the gamma rays are coming from the nuclei, as you know. But something curious happened there, and is related with those applications we are talking about here. That this beta radiation could be positive or negative. Not only electron could be present in this radioactive emission. Also, the positive electron that you know is the positron. And the positron is the antiparticle of the electron. And as happened with all particle-antiparticle pairs, when they meet in a place in the space, they annihilate. And this is an important property we are going to show here in this seminar. Concerning this medical application, since 1951, these radioactive species, nuclei, were used very commonly. The most used was cobalt-60. Cobalt-60 is a beta emitter, but the daughter, which is nickel-60, is produced in an excited state, so emitting some gamma rays with energies of 1.17 and 1.33. Here you have the spectrum of emission of the cobalt-60, where you can see these peaks corresponding to those gamma rays. There is another one there with a question mark. Which one is that, you know? 0.51111. It's a question to the audience. Never heard about it? Do you remember what is the mass? The mass of the electron? 0.511 MeV. So this is the annihilation of electrons and positrons into the detector. Since the cobalt-60 is emitting 1.17 and 1.33, they can produce pair electron-positron pairs, and the positrons annihilate into the detector. It's curious, but it's related to this thing we are going to explain here. This is the typical, this was called, a really bad name, was called cobalt bomb. And was very useful in radiotherapy in the 80s, last century. Very common, but very complicated to use because you can imagine that it's a radioactive source with a really big activity. So all topics related to security and so on, very often. A few years later appears this machine, the linear accelerator. This is a machine that is producing X-rays, similar to that of Röntgen that I show, but the voltage is much higher. It's typically between 6 and 10 MeV, capital M, mega electron volt. And the accelerator is not a simple voltage. It uses an accelerated waveguide to go in phase, but finally the effect is the same. So the some electron with an energy of 6 mega electron volt are stopped by the target, and they produce X-rays. So this machine is producing X-rays by Bremsstrahlung. That's the technical name of the decelerate the electron, Bremsstrahlung. Well, these are the machines that are used for radiotherapy in about 80% of cases. This is the machine that is used for radiotherapy. So we are talking about using radiation, electromagnetic radiation. That's, as you know, at this energy can be treated or considered sometimes like particle beams. And the particles forming the radiomagnetic radiation are the photons. So I will talk about photons. Every time I say photons, photon beams, I'm talking about radiomagnetic radiation, but with a particle behavior. No problem with that. How the medical doctor controls the radiation used in radiotherapy. The reference parameter the reference magnitude is the absorbed dose. And the absorbed dose is the energy per unit of mass absorbed by the target. So it's an absolutely macroscopic unit. And it's the only parameter at the moment that they use for decide a treatment. The next question is how medical doctor or radiophysicists can measure the dose that they are delivering to the patient. What they use is a kind of phantom. This is a phantom of water. It's a simple box with water inside. And is it possible, is a question, is it possible to measure directly the energy absorbed by the water? The answer is no. With the physicists, we don't know how to measure the energy into a body. It's almost impossible. What they do is to measure the ionization induced, the number of positive charge produced by the radiation. And this is something that we really can measure, and we know how to do it very precisely. And then they had to assume that the energy necessary to produce a per ion electron is a constant. This constant is W, which is constant or not. It's almost constant. And multiplying the number of ions by the constant, I get the energy absorbed. So as you see, the way to control the radiotherapy is quite macroscopic. There are no details into the real interaction of the radiation with the medium. The ionization is measured with these kind of devices, ionization chambers. This is a very small ionization chamber that we can introduce into this phantom of water, and this can control with mechanically with a computer. So you know in principle the dose delivered in any point of this phantom. Then this is introduced into a computer, and the treatment is planned in base of the energy deposition and some geometrical consideration like the size, volume, collimators, etc. Okay. Finally, the treatment is there. In 2000, a group of scientists in different places in the world, Canada mainly, in Europe, United Kingdom, I was involved in this initial collaboration as well. There was an experiment showing that not only damage is produced by ionization. Radiation can produce damage at the molecular level, even if it's not ionizing. Some electrons with energy below the ionization level can break molecules. Breaking molecules in some sensitive molecules or sensitive structures like DNA, breaking some of the DNA bases means that we are producing a lesion that could be finally a biological lesion and could be finally a lethal lesion. So this means that the radiation, the effect of the radiation is produced at the molecular level. And this is the subject, has been the subject of our investigation for more than 20 years. That means that we had to study what is the interaction of these particles. Particles constituting the radiation beam that are mainly photons, but also electrons and also radicals. Initially, in the case I show, initially, the primary beam is formed by photons, but photons interact with matter by mainly by three channels. One is the quantum scattering, which is changing the direction simply and transferring some energy, but the photon is still there, but following other direction. The most important for this application is the photoelectric interaction, in which the photon is producing a secondary electron, a photoelectron, with an energy almost the same as the energy of the primary photon. In fact, is the energy of the photon less the energy, the binding energy, which is a few eV only. And the pair creation, sorry, my multiplexer is working not properly, and sometimes it's changing from one language to other language. Pair creation, which is producing positron electron pairs, and very important in this application. But now we go to the secondary particles, electron. These are our main goal, electrons, because the complexity of the electron interaction is much higher than the complexity of the photon interaction. It's not well known. There are many channels open, and we need to know for all those channels, elastic scattering is always present, and we need to know the probability of these elastic scattering processes. I will show you why. Then excitation of the molecules. The excitation could be vibrational excitation, rotational excitation, electronic excitation. There are many channels open. Ionization, of course, which is the very way of producing damage, and dissociative electron attachment. Dissociative electron attachment is our flag, we can say, because it's a process in which energy is not transferred to the molecule. Could be almost zero, but the molecule can be dissociated. The way to dissociate is forming a negative, a temporary negative ion, and finally, this negative ion decay into different fragments. Could be negative, positive, no, not positive, it's more complicated, negative or neutral. And these fragments are radicals, and these radicals are very active and can destroy the basis of the DNA. So we need information about all these channels. This information is in terms of the probability of the process, which is called the cross-section, and also in terms of the angular distribution of the process. Why? Because finally, our goal is to introduce all this data into a modeling procedure, into a big modeling program to simulate what is happening when you irradiate with a high energy particle radiation, what is happening into the target, in this case, into water mainly. We are assuming that the patient is mainly water, but sometimes you need to introduce some molecules like radiosensitizer or other kind of molecules. You can also put it into the model. I'll show you the model in a few minutes. This is the model that we developed many years ago. It has three parts. The input data, so we need to collect, to measure, or to calculate all the input data required by the model. Then the model itself is a Monte Carlo simulation code that we developed many years ago. Our code is called Low Energy Particle Track Simulation Code, mainly focused or developed for electrons, positrons, and protons, charged particles mainly, but using the architecture of other general purpose Monte Carlo codes, like GM4. Do you know GM4? Have you heard about it? No. GM4 is used by all the high energy physics community, was developed at CERN, and it was quite popular. It's not easy to work with it, but we'll say that the applications are really standing out and well documented. So we use the architecture of GM4, and using GM4 only for the high energy, because this is a code that has been developed for high energy physics mainly. Gamos is an interface for medical application, was developed at CMAT 10 years ago, something like that. But we integrate Gamos into GM4, and the LEPS code is also integrated into the other two. So finally, we can simulate the track of any particle, starting mainly with photons, until the final thermalization in the field, in the target, and following the tracks of all the secondary particles created along the track. Okay, this is what we try to do with the model. But a model is nothing if you can't validate. So the third part of this study is the validation, the preclinical validation, to see if finally the biological effect or the medical effect of our model is correct. Our prediction is correct. So this, I'm going to very fast in this part, because this is to describe all the system that we have, all the procedure that we follow to get this information. And I think this probably is not very interesting for this seminar. I go very fast, but if you want to ask anything, then you can do it, and anytime. If you want to visit or to know more about any of the procedure that I'm presenting here, just ask me. So this is very easy because it's the available data in libraries. In fact, is the database for GM4 and Gamos. Nothing to do with that part, just to take it from the web. The source geometry and emission spectra, this is something that you have to measure. But for most of the accelerator used in medicine, it's complicated, no, even impossible to measure directly the spectrum. What we had to do is to simulate the spectrum, which is relatively easy to do it with Monte Carlo techniques. We simulated the spectrum for a LINAC accelerator for 6 MeV, and you can see the shape. The shape is that the maximum energy is 6 MeV, but only a few photons have that energy. Most of them, the maximum is around 1. In fact, the average is 1.82. And this is the energy used, this is the accelerated energy used for more than 90% of the treatment. Why? Because the average energy is pretty close to the cobalt-60, and the cobalt-60 is the source used for calibration. So they can maintain all the dosimetry of cobalt-60 in the case of using LINAC. Interaction cross-section data. This has been my main goal for many years. We have to measure the probability of interaction, not of photons, because the photons are well known, of electron and other secondary particles. In the case of electron, we had different systems at CSIC. This is electron beam generator, which is magnetically confined, and this confinement allows to measure the probability of interaction, the type of interaction, producing elastic or inelastic, and also giving information about the angular distribution. In the case of positron, we have a similar apparatus to measure the cross-section, the probability of interaction, and also this differential cross-section, angular distribution. But this is a bigger, more complicated. It's working with a very intense radioactive source of sodium-22. So it's not in Spain because it will be very complicated. It's in Australia, and we have been working in collaboration for many years, and we are still working with that machine. In the case of radicals, this is one of the machines that we have is our design and construction directly that we have at CSIC there here in Madrid. This machine is producing some kind of radical that are very interesting to study radiation damage. Are producing oxygen radicals. These are very active. We designed this, you see, this is the discharge. Through a discharge, we produce a negative beam that we accelerate and we cross with the gases that we want to study. All kinds of molecules to see what is the reaction of the radical with the molecules. Angular distribution function. This is something that you can measure in principle. You collide an electron with a molecule and then study the direction of the scattered electron. You can get information about the energy and you can get information about the angle of the scattering. This machine is not here, it's also in Australia, but we did all the measurement that we needed in collaboration with Flinders University. It's impossible to measure all the data that we need for the model. So we have to use some calculation. So we developed some calculation method allowing to calculate differential and integral cross-section, so probabilities and angular distribution of electrons with all kinds of molecules. It's not very accurate if you want to know what is some details of the molecules, but it's really, really accurate within 10%. If you don't care about these details and you incorporate those details from the experiment. Also, this calculation method allows to introduce condensation effects. The media that we are treating normally are liquid or solids. And the data that we get from this experiment are for single molecules. Then you have to simulate what is the behavior of that molecule into the model, the surrounding effect of all the molecules surrounding that one. And this is something that we do with this calculation method. So energy loss distribution function is something that we really need to get what is the energy transfer to the medium. And we have another system here in Madrid that allows to measure the energy loss by electron with different kind of molecules, always designed by us and constructed at CMAC. The as I mentioned, is a Monte Carlo code by the general purpose that is given by GM4 and our code, which is close to it. Every time GM4 decide that a secondary particle with lower energy is produced, it enters into our LEPS code. So this part of the code is for protons with less than 1 MeV, positrons with less than 10 keV, electrons with energy length less than 10 keV, and radicals that we are implementing at the moment. For higher energies, GM4 and gamma solve the problem. And finally, yeah, this is just to show you some example of the simulation. This is the water phantom that I show you, and we try to simulate all the processes into this phantom. This is the result. I'm not sure if you can see this. So this is the phantom, 50 centimeter by 50 centimeter by 50 centimeter, and this is the photon beam. As you can see, the photon is crossing almost all the phantom. Even some part of the photon is going out of the phantom or on the bottom. You don't see to see it. A view from the top, we can see the irradiated area. In this case, is a square 10 centimeter into the phantom. But if you see the detail of this region, not easy you can see it, there you see the track of some secondary electrons. And in the case of photoelectrons produced by these photons, the range could be something like 13 millimeters. So you can see that the damage of the radiation that is mainly produced by the electron could happen out of the irradiated area. This is the region of penumbra, and it's something that we have to take into account in the treatment. Not only the energy, the total energy delivered. We need to know what those electrons are doing, what damage are producing, because in that area, there could be other organs that we don't want to irradiate and we don't want to produce damage. If you go to the end of a track, you can see that our LEPS, the previous simulation is the simulation given by GM4. But if you go to the end of the track, you can see that our LEPS code gives information about the type of interaction, the number and type of interaction. There are some elastic. In this case, the size of the balls indicates the energy transfer. And the color of the balls indicates the type of interaction. You can see this blue small is elastic scattering. No, only a few EV could be transferred, or even less. Vibrational excitation is bigger. The molecule is excited vibrationally and even can be broken by vibration. But for neutral dissociation is a clear case in which the radiation, the electron, is dissociating a molecule. So it's producing damage at the molecular level. Then we have other processes like ionization, or electron attachment that I mentioned. Electron attachment is the prototype of process producing damage, not transferring energy. Also, our code provides data. The output is the number of processes of each kind of process. So what we needed to do is to introduce this information into the dose planner of the hospitals. But we are quite far from this point because it's difficult to come. The medical community has medical protocols, and to change the protocols is really complicated. It requires some verification, a lot of verification, and it is something that we are doing now at the moment. And finally, radiobiological experiments, preclinical conditions. We do this in collaboration with some hospital in Madrid. Ramón y Cajal, initially La Paz, now is not working with us, but Ramón y Cajal and Puerta de Hierro. This experiment was done at Puerta de Hierro with some cells. In this case were Jurkat cell, which is this kind of lymphocytes. It's a kind of lymphocyte. We grow the cells into some well and then irradiate it at the different part of the irradiated area, as I showed you. There are 12. The line is formed by 12 wells, and you see the position is this, the irradiated is the center of this part, and the result is this one. The number indicates the position of the well, and you can see that the energy, the position is mainly for the first one because it's the center. Then in this area is the penumbra. There are also energy deposition by this secondary electron dimension. But when we compare with the dissociative electron attachment number, we found that there are not big differences. So we concluded that those measurements done by the hospital when they are using photons, as is this case, is correct, almost correct. The difference when you compare with the radiation damage at the molecular level, taking the dissociative electron attachment as a representative of this damage, is not big. Although we found, studying the damage induced to the cells, that the difference could be important. And out of the irradiated area, many cells died. What we measure here is the apoptosis at different times. Many died out of this irradiated area. So some radical diffusion, some secondary electrons were going there, producing biological damage, although the energy transfer is not very important in these areas. This is something that we are studying at the moment. Okay. It's possible to be more precise using photons. The answer is yes, but with some consideration. This is the CyberKnife. It's the most precise X-ray photon irradiator that are at the moment. This is from the Hospital Ramón y Cajal of Madrid. And what is the important point of this machine is that it's controlled by a computer. It's like a robot. So the radiation can be delivered following different rays, different direction, and concentrate all the radiation into the tumor. It allows to treat lesion on the trigeminal nerve, which is quite delicate, is there, by changing the direction of the beam and crossing all the beam, at the point that we want to irradiate. But of course, it's producing damage around. Less, but some damage is produced. Well, what could be the solution for the future? Well, charged particles are really different than photons. The way that charged particles interact is very, very different than photons. The charged particle is delivering only a small amount of energy and is losing the energy. The photon with a photoelectric effect disappears the photon, and all the energy is transferred to the secondary electron. But a charged particle, electron, positron, proton, whatever, in each collision, no matter what is the incident energy, could be very high energy, the incident, but the energy transfer in each collision is always low, around a few eV, tenths eV, maximum 100 eV. That means that the charged particles have a range into the body. The yellow arrow represents photons. So photons pass through the water and the DNA, a mix of DNA and water, which is what we normally simulate with our model. And they interact, they have several interactions, producing secondary electron, and then pass because the probability of interaction is very low. And most of them are not interacting at all, so pass through the body. And that's the reason why photons are producing damage in other areas. While charged particles are losing energy, so the average energy in the initial could be several hundred of MeV, but finally they stop into the target. So finally, all the energy is delivered. And when the energy is lower, much lower, around, imagine if the initial energy is 250 MeV, when the energy in water, when the energy is around 80 keV, is when the cross-section, when the probability of interaction is higher. And the energy is mainly delivered at that point. That means that using charged particle, controlling the incident energy, we can control the depth. We can control where is totally stop the radiation. And this is the main advantage. And that's why radiotherapy is going these days to use accelerators for charged particles, electrons, protons, heavy ions, carbon, oxygen, or even helium, okay? I don't know, 10 minutes? No, 10 minutes? 10 minutes. I'm going to really fast explain that, but it's one of the key points of my presentation. So electrons, the problem of using electrons is that they don't penetrate much into the body. In fact, radiotherapy using electrons has been used mainly for skin cancer. But now there is a new technique called intraoperative radiotherapy with electron that combines surgery with the irradiator. This is similar to the LINAC accelerator. It's a LIAC because at the end, the beam is an electron beam, not a photon beam. It's not, we don't stop the photon, we don't stop the electron beam, but the electron beam is going out the accelerator and you irradiate the patient with the electron beam. So you need to combine surgery to expose the tumor to the beam. But it's very effective, and it's one of the applications that we are studying with more interest now at the moment. In fact, when we simulate this 6 MeV electron, remember 6 MeV is like a magic number of the medical application, like 6 MeV for the accelerator. The range into water is 35 millimeters. At the end of an electron track, as we showed before, we can predict the number and type of interaction that take place. So to compare the prediction of the model with living cells, what we did is to irradiate these cells. This is a flash sample. In this case, we use epithelial cells because are more easy to control the geometry than the previous one. And we irradiate in 3 points, at 3 different points. One close to the surface, which is the point at which the tumor is placed in a real application of radiotherapy, and then at lower energy of the beam, even at the end of the tracks. Here you have. This is the energy deposition. The maximum energy is deposited at the first point. Then the intermediate point, the energy is almost half. And at the end, the energy transfer is much less, much less. When we studied the survival fraction, in this case, studying the formation of colonies, we got this result. Position 1, the survival fraction is this one, and you can see that for the position 3, where the energy transfer is clearly less, the dose is clearly less, the damage is higher. With the same dose, you know, with the same dose. That means that we irradiated this position much longer to get the same dose. So this relative survival fraction lower means that the effect of the radiation is higher at that energy. And not only dependent on the dose, also depends on the type of interaction and the dissociation, molecular dissociation induced. Bien, well, with protons, the situation is similar on electron, but in the case of proton, it's much more effective because you can accelerate a proton to a very high energy, so you can control what is the range of the protons. And this is the base of the hadron therapy, which is one of the most promising techniques now. Here, again, we compare irradiating with photons. So the photon beam is crossing the patient. And here is irradiating with protons, where you can concentrate the energy deposition mainly into the tumor, because photons are losing energy continuously. Protons are mainly losing the energy around this peak when they get an energy lower than the incident, much lower than the incident. I said about, if the incident is 180 MeV in this case, I say about 80 keV, kilo electron volts. Tuning this incident energy, I can control the depth. So we can put the energy deposition into the tumor. The problem of the hadron therapy is that requires a big installation, big accelerator, are very expensive. In Spain, this technique has been implemented only a few years ago. I will show you one. In Europe, many years ago, I would say 20 years ago. And what we did in the case of proton, we did similar experiment that we have done with electron, but for proton, we had to go to a proton accelerator. In this case, we did the experiment at CEMAN. CEMAN is the proton accelerator of the Autónoma University. This is where we place the cells to measure the damage. And this is the first proton therapy installation in Spain. It's private from Quirón Salud. There is another one, this is in Madrid. There is another one in Madrid. And now there are 10 new ones in Spain, 2 in Madrid, and other part of the country that had been donated by this, the owner of Zara, you know. But these are not working at the moment. They are in installation. Other places in Europe, this is the proton therapy facility of Quirón. Other centers like Heidelberg, 2 more centers in Germany, 3 in Italy. They use more heavier ion, no proton, much heavier ion, for which the effect of this control energy deposition by controlling the energy, the incident energy, is much more clear. But it requires really big and expensive installation. And in Spain, we don't have any. We will see in the future. We have been collaborating with Heidelberg from the beginning of the installation of this one that you see. This is what we call the gantry. This is the way to rotate the proton beam produced in this cyclotron. Well, just a few minutes about the dark side of this point, which is using positrons, as I mentioned at the beginning of the talk. Positrons are used in medicine because the annihilation of the positron produces 2 photons or 3, depending on the total angular momentum. And detecting this photon, we can see where the annihilation took place. And this is important because if we attach a positron emitter, in this case is fluorine-18, we attach that to a molecular marker. In the case of cancer tumor, typically are sugar, like this one, fluorodeoxyglucose, and you attach the fluorine-18 to the molecule, and you introduce that into the body of the patient, the molecule, the sugar, is going to the part of the body where the sugar is demanded. And all incipient tumor and metastasis are demanding a lot of sugar. So when the positron go there and find an electron, they form what we call a positronium. The positronium is an atom formed by electron and positron. Each one rotate around the other because they have the same mass, so it's like a dance. And finally, they annihilate, producing two photons. Those photons can be detected by these gamma ray detectors, and the computer is telling us where is the point where this annihilation took place. In the case of our model, we can predict the final track of a positron. This is a picture that I show many times because it's really illustrating the situation. It's the end, the very end of the track of a positron. So positronium is formed here, is the red ball, but before is annihilate, OG electron is produced, and a new track is generated, and we could simulate the track of the secondary electron. And finally, by electron attachment, is dissociated in the water molecule. Okay, in combination of this technique, combined with CT, CT is sometimes here is called TAC. Combining TAC and PET, positron emission tomography, you can get very clear images or all the future of some incipient tumor or metastasis. Here is an example. This is a real case. Different slices of the body or the scanner. Every time you see a point, illuminated point, it means that the annihilation of positron there is taking place very frequently. So it's indicating that some lesion is there. You follow all the body. And finally, the diagnostics in this case was primary pancreatic tumor with several hepatic metastases and incipient pelvic and cervical lesion. It's probably the most powerful tool for diagnostics of cancer and other illness. Because depending on the molecular marker that you use, that is the effect that you are detecting. In the case of Alzheimer, can be used this technique as well, but uses beta amyloid, which is a protein goes to the body. So finally, my acknowledgement to all people who have collaborated with this project. This project is finishing, in fact, finished. And the people who work now are distributed into different groups, and they are still working. And I'm planning to continue helping them and supporting them before my retirement will take place in only a few months. But remembering some of those, in particular, I would like to acknowledge the help of the student, that now all of them are very important people in the field of science. And thank you very much for your attention. I'm sorry for the multiplexer, but it's something that happens with age and with lack of practice.
Well, thank you very much, Gustavo, for that fantastic talk. It's fascinating to see some of the applications of what you're doing, and the sort of images at the end, and the diagnosis at the end kind of brings it all home, right? How important this work is. Yeah. So if anyone in the room has questions, please raise your hand. And equally on Zoom, if anyone has questions on Zoom, please raise your hand.
No question.
Well,
about the Monte Carlo code.
I was interested about the timescales, and you mentioned it a little. I think maybe at the end.
On the positron.
Yeah, of timescale of thermalization.
Well, thermalization. Yeah, timescale to the final thermalization of the electrons. It could be, yes, nanoseconds. I haven't talked about the time, but the only time detail I gave was about the life of the positron. It was very fast at that moment. I didn't say anything, but it's really is really fast. So the life is very short. at the positron in the body is just 10 to minus 7, 10 to minus 10 seconds. Positron. But the total drag from the beginning depends on the energy of the initial electron, but could be on the order of microsecond, nanoseconds.
Of course, I have talked with you before this talk. So one of the worries is the damage of the radiation that is not controlled, in the sense that it's not on target, but it is spread on cells outside the tumor, for instance. But now I am worrying about this positron, this PET tag for the detection of the tumors, because then the 2 or 3 photons that you produce from the positron are gamma rays, and so much more energetic than the X-rays. So is it very low dose of gamma rays that is coming out? Because in principle, this is also quite damageful, no?
Well, when we talk about damage, I'm giving details, very precise details about the damage produced at the molecular level. But when you talk about real treatment, the accuracy is not so precise. Why? But it's normal, because medical protocols have to follow what is written in the protocol. So if everything has to be proportional to the dose, everything is proportional to the dose. So the treatment is based only on the dose. Radiotherapy, I'm talking. If you want to know what is the radiobiological effectiveness of that radiation, the protocol gives some factors to multiply the dose. In the case of using protons, the factor is 1.1 or 1.15. You multiply the dose by that factor, and this is your biological effect. So your question is about the detail of dosimetry of PET. There is no dosimetry. Only they treat that the radioactivity of the fluorine-18 was not too big, not too high, to produce any damage into the body. But the possible damage of the photon is not accounted for. If your question is, can be calculated, can be modeled, the answer is yes. But is it going to be used for a real PET? Probably not. Probably not. Even if when you go to the medical doctor, you have X-ray, radiography. Is any control? Is anyone controlling the absorbed dose? They follow the protocol. And the protocol say, if a person is just receiving this dose of radiation, there's no problem. More dose could be problem. So not go into that region. No more than one tomography per month, or 2 months, and that's the protocol. It will be really complicated to evaluate what is the real damage and the biological damage, not to say. I showed some of the biological results. Did you see the error bars? About 20%, 25%. For a physicist, this is terrible, but for a biologist, when you talk about biology, it's normal because they follow some statistics measurement, and this is the statistic. It's not possible to reproduce that with better accuracy, at least for us. Okay.
Thank you very much for the nice talk. I was wondering whether your Monte Carlo code could be applied to other fields, like astrochemistry, for instance, because some of the processes that you mentioned there, we believe happen also in interstellar ice, or whether that could be applied to the surface of icy moons, or to estimate how much material, biological material could be damaged, or whether there may be any tracks of that radiation happening.
This is a very good question and a difficult answer. But when you say Monte Carlo code, when people say Monte Carlo code, in fact, you are saying almost anything. Because Monte Carlo is a mathematical form to treat random number, to reproduce some statistics data that you introduce into the model. So the answer in principle is yes. With a Monte Carlo, you can simulate anything you like. The question is, do you have the data that you need to simulate that precisely? And that is the question. In that case, you had to go into the data to see what is the origin of that data. When I show GM4 is for high energy physics. And it's using the data available in libraries like Livermore, some of them. If you go there, you will see that that data is only applicable for really high energy because most of them are based on the Born approximation. So if you use that to simulate something that is happening with very low energy electron on a surface, with a big molecule or a film of molecules, the answer is you can do it because with a computer, you always get an output. It's a good thing of the computer. With experiments, sometimes you don't get anything, and there's nothing, no? But with a computer, you always get an answer, an output. But this output is reproducing what is happening there, so it will require some validation. First, go into the data to see if the data correspond to your problem. And even in that case, then you have to validate reproducing some. We are doing that with these living cells, but in your case, you have to measure all the indicators that, I don't know, association, radicals coming from the surface or whatever. But in principle, with a Monte Carlo, in fact, Monte Carlo is called, the name Monte Carlo comes because the first application was for casinos. To predict in a casino the result of random processes. Like that.
Any further questions here in the room? And I'll give a final opportunity for questions on Zoom. Then if not, let's thank the speaker again for a really interesting talk.
Thank you. Thank you.
I really was an interesting talk. You know.